Hi, my name is Erin Klostra, and the date is, you can see that, December 18 of 2022. And this is my presentation on the determination of gender, specifically the roles that genes and hormones play in that determination. So, each human is programmed toward sexual development with sex chromosomes. There are 23 pairs of autosomes and one pair of sex chromosomes, which are either XX or XY, depending on the sex of the human. Each egg produced by a female has an X chromosome, and each sperm produced by males has either an X or a Y chromosome. So females will inherit one X chromosome from each parent, and males inherit one X from their mother and a Y chromosome from their father. The diagram illustrates how sex is determined according to which pair is formed. So, while the autosomes determine the direction of many bodily characteristics, the sex chromosomes determine the development of the internal and external sex organs, as well as the secondary sex characteristics. The Y chromosome is imperative for development of the male reproductive organs. They can't be formed without it. So, to start off with sexual differentiation, the embryonic tissues begin with a bipotential gonad, which you can see at the top of the screen in the image. And up until the 7th week of development, they have this bipotential gonad, and then between the 7th to 10th week is when they begin to differentiate. So it's called the bipotential gonad because it cannot yet be identified as either male or female, and the internal sexual organs begin to form around 10 weeks. So if the gonad is to become an ovary, it will do so in the absence of the SRY gene. You can see this on the left side for female development, and it will do so in the presence of some female-specific genes. The absence of testosterone causes the Wolffian duct to degenerate, and at birth, the absence of anti-Mullerian hormone in the female infant will allow the Mullerian duct to become the fallopian tube, uterus, and the upper part of the vagina. On the other hand, if the gonad is destined to become male, like on the right side of this diagram, the SRY protein will direct the medulla of the gonad to become a testis. The newly developed testis releases anti-Mullerian hormone, which causes the Mullerian ducts to disappear. So once the male infant is born, the testosterone produced from the testis converts the Wolffian ducts into a seminal vesicle, epididymis, and vas deferens. So while there is hormonal influence, sex determination primarily depends on the sex-determining region of the Y chromosome, also known as the SRY gene. Presence of the SRY gene creates masculine structures, and absence of the gene creates feminine features. So as was mentioned in the previous slide, the bipotential gonad begins to differentiate between the 7th and 10th weeks of development. The figure displayed on this slide shows the development and differentiation of the external genitalia, which comes from the genital tubercle, which is shown on the first picture on the very top of the slide. So if the bipotential gonad is to become female, the genital tubercle will form the clitoris, the urethral folds, the grooves will form the labia minora, and the vaginal opening, and the urethra, and the labioscrotal swellings will form the labia majora. So this feminization of the external genitalia happens in the absence of androgens, as we kind of had talked about before. And on the right side of the image, we see what happens when the bipotential gonad is masculinized. So in this case, the genital tubercle forms the glans penis, the urethral folds and grooves form the shaft of the penis, and the labioscrotal swellings form the shaft of the penis and the scrotum. This masculinization of the bipotential gonad occurs in response to the presence of dihydrotestosterone, also known as DHT. When the male infant is born, the testes descend from the abdominal cavity into the scrotum, which is shown on the bottom right image. So this is just sort of a little diagram I put together to make sense of the hormonal determination. So the hormonal influence on gender development occurs in the presence of SRY for males and the absence of SRY for females. The diagram here shows the influence of the SRY gene on male development, since female development is much less hormonally regulated and has more regulation by genes in development of the ovaries. So first, the SRY gene produces testes-determining SRY protein, and then this protein initiates the production of several other proteins that cause the gonad medulla to differentiate into a testis. And then the testis has both interstitial and Sertoli cells, which play different roles in development. The interstitial cells secrete the hormone testosterone, which controls the development of the Wolffian ducts, as well as the development of the external male genitalia through DHT. The Sertoli cells, on the other hand, secrete anti-Mullerian hormone, which causes the regression of the Mullerian duct to inhibit feminization of the organs. Most other sex characteristics are controlled by DHT. So the differentiation of sexual organs in humans is highly integrated between genetic and hormonal influences, as we've already seen kind of the interplay of those. But dysfunction in this interplay, I think, especially highlights the importance of genetic and hormonal coordination, and it's seen in several disorders such as congenital adrenal hyperplasia and pseudohermaphroditism. So first, congenital adrenal hyperplasia is a recessive autosomal disorder which results from mutations of the CYP2A12 gene, which causes inappropriate elevation of specific adrenal hormones and deficiencies in others. This leads to an increase in overall testosterone, which leads to development of various degrees of virilization of the female genitals. And then pseudohermaphroditism is a condition in male humans that involves different genes and hormones but leads to a similar outcome as the first disorder we just discussed. So males with this condition will inherit a defective gene for 5-alpha reductase, which is what catalyzes the conversion of testosterone to DHT. And we've already kind of talked a lot about the importance of DHT, but since it can't be formed, the male external genitalia and prostate gland fail to develop fully and normally in the fetal stage. So in this situation, the human will appear female at birth and will be raised as a female, but will inherit many masculinizing characteristics like masculine external genitalia and pubic hair when the testes begin to secrete testosterone, but this won't happen until puberty. These are just two among many different disorders that can be discussed concerning the interplay between genes and hormones and sexual differentiation, but I think that they each demonstrate the importance of each influence in successful masculinization or feminization. And then this is just a diagram. I thought it lays out all the important genes and hormones of sexual differentiation. So the genes are in bold, and then the hormones and some of the accessory ducts and things are all boxed in this diagram. So now that we've seen what can go wrong with this process, I think it's important to highlight all the steps that need to happen in order to make it successful, to make it go right. So I thought it worked well as a study tool. I think that it just lays out everything in a way that makes sense, that's easy to grasp. It's a tool I find helpful for studying since I like to draw these diagrams and give myself a word bank to put the diagram together in the proper order. And I think it helps to visualize the outcomes and effects of each gene and hormone and how they affect each other and the overall effects they have on the body. So when I've done this enough times, usually I take the word bank away and I try to draw this from memory. So, and then here are my sources. Thank you.